This is Mathieu Lupien presenting a discovery note on my team's latest work published in Cell Stem Cell this November 2020. The work is entitled The Transition from Quiescent to Activated States in Human Hematopoietic Stem Cells is Governed by Dynamic 3D Genome Reorganization. The work was led by Drs. Naoya Takayama and Alex Murison and conducted in collaboration with Drs. John Dick, Stephanie Chi, Juan Vaquerizas, Michael Taylor, and Danielle Di Carvalho. Each day, our body produces over 1 trillion blood cells. These include mature cells, such as platelets and T cells, that originate from the differentiation of hematopoietic stem cells into one of many progenitor populations. Two distinct hematopoietic stem cells are known. These are the long-term and the short-term hematopoietic stem cells, also known as LT-HSC and ST-HSC, respectively. Long-term hematopoietic stem cells lie at the apex of the hematopoietic lineage and can give rise to the short-term hematopoietic stem cells. While both can differentiate to give rise to all needed downstream progenitor and mature blood cells, there are important differences between long-term and short-term hematopoietic stem cells. For instance, long-term hematopoietic stem cells take their name from their long-term ability to self-renew, an essential feature allowing for blood production throughout our lifespan. In contrast, short-term hematopoietic stem cells have reduced self-renewing ability, which leads to their depletion over time. Hence, short-term hematopoietic stem cells are primed to differentiate. This is reflected in the ability of short-term hematopoietic stem cells to exit dormancy, also known as quiescence, much faster than long-term hematopoietic stem cells. By exiting quiescence, hematopoietic stem cells enter the cell cycle and differentiate. Long-term hematopoietic stem cells can therefore be seen as a reservoir for blood cell production. In contrast, short-term hematopoietic stem cells are primed to respond to the need for rapid blood cell production. We therefore decided to investigate how long-term hematopoietic stem cells maintain their long-term ability to self-renew and how they control their commitment to becoming short-term hematopoietic stem cells. Our efforts therefore focused on identifying the determinants of stemness. Knowing that the genetic contributions to phenotypes are entrusted to the chromatin, where the chromatin serves as the scaffold of genetic architectures, we decided to investigate the nature of chromatin accessibility and compaction across both long-term and short-term hematopoietic stem cells, as well as 4 progenitors and 6 mature populations. Our goal was to identify distinct paths of differentiation from long-term to short-term hematopoietic stem cells embedded within the chromatin accessibility profiles of the most primitive to the most mature hematopoietic population. Comparing chromatin accessibility profiles across all hematopoietic populations identified 10 signatures, representing paths to differentiation. Some were specific to unique mature populations, such as for the T cells or the NK cells. 2 were shared across stem and progenitor populations, namely the LT-HSPC and the activated HSPC signatures. Noteworthy, the activated HSPC signature was not enriched in long-term hematopoietic stem cells, while the LT-HSPC signature was enriched across both long-term and short-term hematopoietic stem cells. Scoring the LT-HSPC and activated HSPC signature across single-cell ATAC-seq data generated in a mix of stem and progenitor populations defined by their CD34, CD38 cell surface markers reveal the enrichment of the LT-HSPC signature in green in the CD34 positive, CD38 negative populations, a population known to be enriched for stem cells. A gradual transition for the enrichment of the activated HSPC signature in yellow could also be observed towards CD34 positive, CD38 positive populations, commonly assigned to progenitor populations. The LT-HSPC and activated HSPC chromatin accessibility signatures were next mined for DNA recognition motif enrichment, with the intent to identify DNA binding proteins differentially active across these signatures. DNA recognition sequences for CTCF, a chromatin looping factor, were found to enrich specifically in the activated HSPC signature, suggesting that gains in CTCF activity on the chromatin could discriminate long-term from short-term hematopoietic stem cells. CTCF is a well-known master regulator of chromatin interactions, where DNA sequences distant from each other in the linear scale of our genome are brought in contact in the three-dimensional space. It commonly sits at the anchor of chromatin interactions, allowing, for instance, to connect individual elements together, such as gene promoters with their distal cis-regulatory elements. Similarly, CTCF is commonly found at the boundaries of topologically associating domains, known as TADs, that correspond to clusters of chromatin interactions. We therefore map the three-dimensional genome organization in long-term and short-term hematopoietic stem cells, adopting the low-input high-C protocol developed by Dr. Vaquerizas to work with a few thousand cells, the number of cells we typically have access to from any individual sample. Overall, our results reveal that long-term and short-term hematopoietic stem cells are fairly similar to each other in terms of TAD structures. This is showcased by the strong correlation in insulation score between these two populations. In contrast, we found a collection of 351 chromatin interactions, also known as chromatin loops, uniquely gained in the activated HSPC sites associated with short-term hematopoietic stem cells and downstream progenitor populations. This is reflected in the weaker differences in normalized aggregate signal at the high C peak versus flanking regions in the LT-HSC compared to the ST-HSC population. Looking at the genes enclosed within the 351 chromatin interactions specific to the activated HSPC signature, revealed their downregulation in short-term hematopoietic stem cells and other progenitor populations compared to long-term hematopoietic stem cells. This suggests that the increased activity of CTCF leading to short-term hematopoietic stem cells and downstream progenitors serves to establish new repressive loops that target stemness genes. In agreement, CTCF knockdown in LT-HSC interferes with their ability to differentiate across both the myeloid and lymphoid lineages. It also interferes with LT-HSC exit from G0, as showcased in vitro. In vivo, silencing CTCF leads to a significant increase in the proportion of long-term hematopoietic stem cells as opposed to short-term hematopoietic stem cells or other progenitor populations, again in agreement with difficulties in exiting G0 of LT-HSC upon CTCF downregulation. Collectively, this work showcases the direct contribution for CTCF in the exit of long-term hematopoietic stem cells from quiescence and entry into the cell cycle required for differentiation for transition to becoming short-term hematopoietic stem cells. In absence of CTCF, long-term hematopoietic stem cells remain quiescent. Hence, CTCF serves to induce the first fold in the genome of long-term hematopoietic stem cells that correspond to the first order fold from a flat sheet of paper one would use for origami. Once folded, it is committed to one of multiple fate, while it remains the reservoir of choice as an unfolded template. With this, I thank you for listening, and I hope that the discoveries presented in this discovery note will raise your interest to delve deeply into the manuscript, now available at Cell Stem Cell. Thank you.